flowering plants reproduce randomly. Flowers are pollinated by insects, or wind, or water. Seeds are dispersed by the same forces of nature. Only the fittest survived until a new force in nature intervened. Man gave up the nomadic hunting life and became a farmer. Now he could cultivate the plants which provided him with food. Through thousands of years of farming, man has begun to understand the growth of plants. The effects of the environment were obvious. Drought, poor soil, and weeds were bad for his crops. Fertile soil and water made them flourish. Yet, having cultured such plants as these Brussels sprouts, man still could not explain why seeds from the same plant, when planted in identical environments, did not always grow into identical plants. Sometimes he overcame this by reproducing plants from cuttings. These always grew identical to their parents and sisters. But plants grown from seeds often appeared different from their parents and their sisters. The cause of this variation was a mystery. Then, a century ago, Gregor Mendel found a clue to inheritance. Examining peas, he discovered a link between generations. His method was to follow the history of single traits in a plant over several generations. An obvious variation between one plant and another was flower color. Mendel took plants that bore red flowers year after year and removed their stamens to prevent self-fertilization. He introduced pollen from pure breeding white flowers onto the stigma of the red. What flower color would the resulting plants produce? Authentic redness had appeared in this, the first generation cross. These red flowers he allowed to fertilize themselves. Before the flowers open, the stamens grow over the stigma and leave pollen on it. What color would their seeds produce in the second generation? Most produced red flowers again. But a few had pure white flowers. Mendel counted his results. He found approximately 3 times as many plants with red flowers as plants with white ones. When he crossed other features, plants which produced round seeds with plants that produced wrinkled or tall plants with short, the same 3-to-1 ratio always appeared in the second generation. 3 round to 1 wrinkled. 3 tall to 1 short. He called the least frequent characteristics recessive, namely shortness, white flowers, wrinkled seeds, round seeds, red flowers, and tall plants, the characteristics which were more frequent, he called dominant. Mendel deduced the basic laws of inheritance. Each single feature, such as flower color, is the result of two character factors. 2 for redness, or 2 for whiteness. At fertilization, one character factor from each parent goes into an offspring. Where these differ, the recessive factor is masked, and the new plant will respond to the dominant factor. This accounted for all of the first generation flowers being red. In the second generation, if one of each pair of factors comes from each parent, there are 4 possibilities. Red and red turn out red. White and red turn out red. White and white turn out white. Redness and whiteness appear in a ratio of 3 to 1. This is the basis of genetics. Today, in the nucleus of a pea cell, we can see something of the material of inheritance which Mendel deduced must be present. The nucleus of every living cell contains thread-like bodies called chromosomes, carrying on them all the information necessary for the transference of characteristics. There are 14 chromosomes in the pea. When the cells are formed for sexual reproduction, the 14 chromosomes move into 7 pairs. The cell then divides. One chromosome from each pair going into the nucleus of each new sex cell. This happens in the formation of both the female egg cell and the male pollen cell. After pollination, a tubule containing the male nucleus grows down the style. In the ovary, the male nucleus fuses with the female nucleus. The newly formed nucleus, with a complete set of 14 chromosomes, contains all the information from which the new plant can develop. How does this chromosome behavior fit with Mendel's theoretical findings? Let's take one characteristic, flower color. Mendel's character factors, now known as genes, still cannot be seen, but we know they are carried on the chromosomes. Suppose a red-flowered pea has 2 genes for redness, one on each chromosome of a pair. When the chromosomes separate, one gene goes into the nucleus of each sex cell. In the white-flowered pea, the same division occurs. When red flowers are crossed with white, each parent contributes one of the chromosomes in each pair. So the offspring gets one gene for red and one for white. Geneticists now know the exact place of certain genes on the chromosomes. For instance, on the 4th chromosome in the pea, one gene controls whether the plant bears many pods or single pods. Further along, another controls plant height. And still another affects the resistance to wilt disease. Present-day knowledge of genetics allows the plant breeder to go through the door which Mendel opened. He can breed new combinations of characteristics, new varieties of plants suited to large-scale food production, short varieties to allow machine harvesting, whole fields maturing on the same day. Breeding for new and better varieties is constantly going on. Varieties which are frost-resistant, with improved taste, bearing more pods per plant, and more peas per pod. One plant already in commercial use is short, resistant to wilt, bears single pods, and produces wrinkled seeds, which are sweeter tasting and suitable for freezing. How could its yield be increased? Another variety is too tall and susceptible to wilt. The peas are round and less good for freezing. But this plant has many pods on a single stalk. Could the best characteristics of the first plant be retained while adding only the multipod characteristic of the second plant? What is involved? Wanted, a pure breeding plant with these four properties: short, multipoded, with wrinkled seeds, and wilt resistant, to be derived from these two parent plants. Let's start with the seed shape. We know from Mendel that roundness is dominant in the same way that red flowers were. So in the first generation, the wrinkle factor is masked. Of the second generation, only one of four will have wrinkled seeds. But we want wrinkled seeds and shortness to breed true. Only one of 16 can grow into a plant that is both wrinkle-seeded and short. Include wilt resistance, and the odds lengthen to 1 in 64. Some characteristics are controlled by more than one gene. Multipodedness is controlled by 2 genes from each parent, 4 altogether. So for multipods, wrinkled seeds, shortness, and wilt resistance to be pure breeding in one plant, the odds are 1 in 1,024. The ideas of plant breeding are easy enough to work out on paper, but the field work is seldom straightforward. A new variety could take 15 years to cultivate, and even then might have hidden drawbacks which could not have been foreseen. The parent varieties are selected and cross-fertilized. The pods develop, and when mature, these seeds are planted. First-generation plants grow. The plants of the first generation are left to fertilize themselves. More time passes before the thousands of second-generation plants are sufficiently advanced for the breeder to start selection. Somewhere here should be the plant with the 4 wanted characteristics. All the tall plants can be discarded. This one looks promising. It is short and multipoded. It is too early to tell which have wrinkled seeds, and further generations will have to be grown to be certain of resistance to wilt. Is the new variety what was wanted? We test for yield and processing, tenderness and taste. If these tests and the field trials are satisfactory, then the large quantities needed for commercial use can be grown in the following years. Plant breeding is a complicated, laborious business which will not be hurried. Today, Mendel's principles are only a fragment of a whole complex of factors which the plant breeder must consider. Yet, present-day crops, when compared with their ancestors, are eloquent testimony to the plant breeder's work. Success in plant breeding comes from an understanding of heredity and environment. We now know that the genes on the chromosomes in the nucleus of a cell are the particles of matter which control all heredity. We can calculate the chance of any one characteristic emerging. Mendel, through his study of the ratios of dominant to recessive characteristics, was the first to consider that inheritance is controlled by physical particles. This was the beginning of the science of genetics.